guys here at Missouri S&T are experts in power systems, and they have fantastic hardware for simulating these power systems. We are going to connect that hardware directly up to live neurons growing on culture dishes in my lab that are instrumented with electrodes. So we have dishes like this one here with 60 electrodes embedded in the substrate, and brain cells from rats, from the rat cortex, are growing on these. They form a two-way long-term connection between the neurons and a computer, and we're able to record the signals that the neurons produce and use those to control some artificial systems such as power simulations. And we can take sensors from those systems and use that as sensory input to the living neurons. So we built closed-loop systems where these neurons talk to external hardware. In previous embodiments, they've been robots, mobile robots, or robots that draw. And the next embodiment will be these power system models running on. So this is an example of a cultured neural network growing on one of these multi-electrode arrays. These are the electrodes, of which there are 60 embedded in the substrate that the cells are growing on. And you can see that after a few days, this is day 3 in culture, they've grown connections to each other. They form synaptic connections and are starting to send signals to one another. And we can pick up those signals, and we can influence those signals, by electrically stimulating them. So, in our lab, we have come up with algorithms for stimulating them in such a way that we can train the network to accomplish certain tasks. So we've had these networks controlling artificial animals called animats. They're driving the animats around in an open field, and we give the animat a task to move in a certain direction. And if it moves in the right direction, we give it a certain kind of stimulation. If it moves in the wrong direction, we give it a different kind of stimulation that will train it, hopefully, to move in the right direction. So we've got different types of electrical patterns that we use to stimulate these networks, and we were able to get them to learn to control the animat to move in a certain direction. So you can see that by day 11, these cultures are very dense, lots of cells, lots and lots of connections between them. The cultures have approximately anywhere from 10 to 100,000 neurons in them, and probably millions of synapses between those neurons. Unfortunately, thanks to the fact that our dishes have only 60 electrodes, we can only record the activity of a few of those. And the big effort in my lab is to move in the direction of optical interfaces. So we are hoping to set up systems whereby we can stimulate the cells with different colors of light emitted by laser beams that are scanning across the networks, and also systems for recording the network activity optically by staining the cells with voltage-sensitive dyes. So we stain these and we image them with a very fast camera that can watch the signals as they pass from one cell to the other.